Chapter 4 is called A Tour of the Cell. It's basically an overview of cells in general and their parts and their functions. Now in Section 1 it discusses microscopes. Before the microscopes were invented in the mid-1600s, nobody knew anything about cells. But it says here, it shows you, it talks about cell theory once again. We discussed this in a previous chapter. But it also shows you different types of microscopes, such as the light microscope and the electron microscope. So obviously the discovery of cells is based on the invention of the microscope, and better microscopes allowed us to discover more. Now in Section 2 it talks about cell sizes. Most of what we know about cells is that they're itty-bitty. Even the bigger cells are still itty-bitty. The reason why cells are small by our perspective is because they need surface area. They rely on their surface area to get in oxygen, get out carbon dioxide, everything that they need and everything they need to get rid of. So the larger it gets, the less surface area it has with respect to its volume in proportion. The smaller an object, the greater its surface area per volume ratio. A good example of this is Reese's peanut butter cups. If you've ever eaten the little bitty ones, you notice that they taste more chocolatey because you've got less volume, more chocolate proportionally than the bigger ones that are used for the Easter eggs. Now, and that limits the size of cells, their surface area. Now again, sections 4.3 and 4.4 discuss prokaryotic and eukaryotic cells. We went over this in chapter 1. Prokaryotic cells do not have a nucleus. They're smaller, they're simpler, they include bacteria. The eukaryotic cells are radically more complex. You have the membrane-bound nucleus and the organelles. So most of the chapter deals with eukaryotic cell structure and function. Now, starting in section 5, you have the nucleus, the most conspicuous part of the cell. It's surrounded by a nuclear membrane. It has our chromosomes inside for our genetic material. It'll have a smaller spot in the middle called a nucleolus, which makes ribosomes. Ribosomes are tiny little dot-like structures in the cell. They make proteins. Now, ribosomes can be floating free in the fluid, or they could be bound to membranes of the endoplasmic reticulum. Well, the endomembrane system is an entire network of membrane-bound organelles that interact with each other. They merge, they separate, not all that different from, say, the bubbles of a lava lamp. And they include the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. Now, endoplasmic reticulum falls into two categories, smooth and rough. Rough endoplasmic reticulum is peppered with ribosomes, and since ribosomes make proteins, that's the function of rough endoplasmic reticulum. Smooth endoplasmic reticulum does not have ribosomes, but it does other things such as make lipids, make cholesterol, detoxify substances, and so forth. The Golgi apparatus is kind of like the UPS of the cell. It sorts and packages and ships cell products from cell part to cell part. Lysosomes are structures that contain digestive enzymes. They merge with other organelles to break down molecules. Vacuoles serve numerous roles. Some vacuoles, such as the large central vacuole in plants, serve to store water. There also are food vacuoles, which hold bits of food. Now, other organelles, such as mitochondria and chloroplasts, are not part of the endomembrane system. They don't interact in the same manner. Now, mitochondria are for processing cell energy. They're the location of aerobic cellular respiration, the conversion of food energy into cellular energy, which is known as ATP. Now, chloroplasts, which are green because they contain chlorophyll, are the sites of photosynthesis. And that's the conversion of energy from solar energy to food energy. So both of these are energy converting organelles. Both of these are double membrane bound. And both of these have their own little piece of DNA in it. And for that reason, section 15 discusses the endosymbiotic theory. That is the idea that the mitochondria and the chloroplasts were at one time free-living bacteria-like organisms that became permanently engulfed and used in a larger cell. Now, the endosymbiotic theory does not explain the origin of the endomembrane system. It's specific to these. Other structures in the cell include the cytoskeleton. a series of proteins, microtubules and stuff, to kind of give the cell its shape, allow for organelles to move, and so forth. Now, these proteins are also used in cilia and flagella, section 17. Now, usually they're involved in cell movement. Cilia are tiny little hair-like structures that may beat in unison to propel a cell in water, and flagella are longer structures. We also have ciliated cells in our body. The cells that line our respiratory tract have cilia to kind of help keep the lungs clean. Sperm cells have flagella. Now, some cells, such as plant cells, have cell walls. A cell wall is a rigid structure that is outside of the main cell or main plasma membrane. Now, the cell walls are made of different things in different organisms. The cell walls of plants are made of cellulose. The cell walls of fungi are made of chitin, different substance. The cell walls of bacteria are made of peptidoglycan, a little bit different substance. However, animals do not have cell walls, and neither do most protozoans. And that's a brief review of this chapter. Be sure to study the notes and the pictures in the book.